Hello, in this video we will discuss about the Tay Sachs disease. Basically, Tay Sachs disease due to the chromosomal defect, the recessive allele both in the chromosome 15. So we will learn about here is the central nervous system and peripheral nervous system. Central nervous system contain brain and spinal cord, while the motor and sensory neurons are present in the peripheral part of the body is known as the peripheral nervous system, a PNS and CNS. So let's draw a brain neuron here, which that's contain chromosome. So chromosome 15 involve containing allele. Both allele is recessive, hex A allele, which that is responsible for the GM2 ganglioside homeostasis in the plasma membrane of the neuron. So the Tay Sachs disease is heritable metabolic disorder commonly associated with Ashkenazi Jew, has been found in the French Canadians of southeastern Quebec. The severity of expression and age at onset of Tay Sachs varies from infantile and juvenile forms that exhibit the differently, and their effect basically the severity of expression and age at onset of Tay Sachs varies from infantile and juvenile form that exhibit. It can be a paralysis due to the neuron dysfunction, the motor nerves dysfunction, and the dementia. Dementia is due to the accumulation of the GM2, the GM2 ganglioside. So the paralysis and the dementia and the blindness is due to the accumulation of the excessive GM2 ganglioside, the lipid, because the ganglioside is the lipid, remember, and the early death and neuron dysfunction and psychosis can be occur. So the Tay Sachs disease in autosomal recessive disease caused by mutation in both alleles of gene hex A. On chromosome 15, so remember that. Okay, hex A codes for the alpha subunit of enzyme beta hexosaminidase A. This enzyme found in lysosome, remember. So the lysosome involving in this mechanism. So again I am repeating, the hex A codes for the alpha subunit of enzyme beta hexosaminidase A. This function found in lysosome, so remember that. Function of lysosome we will discuss now. What is the function of lysosome in this manner? So the break down large molecule for recycling by the cell, and the beta hexosaminidase A. The beta hexosaminidase A will basically lead to lipid. It is normal, will lead to lipid metabolism and GM2 ganglioside. Basically, it is normal. When decrease of the beta hexosaminidase A through absent or decreased due to the allele recessive, and in this way abnormal GM2 ganglioside in neuron accumulation in cell, because it will not homeostatic. So their formula is here, which that's contain D-glucose, D-galactose, and the, as you can see, this is the N-acetyluramic acid and N-acetyl D-galactosamine and D-galactose. This is the combination of the glucose which that is attached with the fatty acid 2 layers, fatty acid 2 chain. So let's begin to understand the mechanism of Tay Sachs disease. So the Tay Sachs disease basically occurring in the neuron, in the nervous system, the central nervous system as well as peripheral nervous system. So this is the soma, axon and Schwann cell of the neuron. This is the cell of the nervous system. And this is the structural and functional part of the nervous system as a cell. Contains cytoplasm and lysosome are present here. This is the plasma membrane, contains bilayer of phospholipid like a simple cell. So the model for GM2 ganglioside metabolism in this video we are going to understand. So this is the attachment of the GM2, and this GM2 is the basically is the ganglioside, GM2 gangliosides, which that is insert into the plasma membrane, but during the excessive will lead to GM2 activator, which that will be the beta hexosaminidase A will be expressed through genes, will lead to attach, and the activator site will lead to GM2 will absorbed by this enzyme, and in this way it will be degrade. The degrade means the alpha and beta subunit which that contain hexosaminidase will bind to supplies this GM2 into the cutting, and in this way the dysfunction. And when the requirement will be happen, so it will again to convert the GM2 to insert for the normal function, but the less amount of the beta hexosaminidase A are absent will lead to accumulation of these lipids into the cytoplasm as well as the plasma membrane. So in this way the complication will arise on the basis of age wise. So the treatment we will discuss, the ganglioside synthesis inhibitor can be used. The ganglioside in synthesis inhibitor means it will be not synthesized, because the synthesis process is the different, we will discuss in the next video. So the ganglioside synthesis inhibitor will inhibit the synthesis of ganglioside because the enzyme is absent. So you should need to inhibit the ganglioside synthesis through inhibitor as a drug. And the treatment combined with the DNA and enzymatic screening programs currently in use will lead to eventually control of this disease. And here let's begin to understand the effect of hexa mutation. Basically the hexosaminidase A mutation, which that will lead to decrease, and in this way the first of all we will understand about the neurons and the neuroglial cell as a macrophage like structure, which accumulation of membrane cytoplasmic bodies, lysosomes containing ganglioside, which that deposition and will not convert and not break down, and the accumulation of complex lipid in macrophage, because the lysosome is basically used for the inhibition due to the enzyme, so lysosome not contain this enzyme, that is why. Accumulation of the complex lipid in macrophage will occur, will lead to dysfunction of the macrophage in the brain, and the destruction of neuron, and the hypotension due to the brain accumulation of lipids, the GM2, the gangliosides, and in this the destruction of neuron, the hypotension will occur, inability, destruction of hypothalamus, and in this way the inability to sit and hold head, and in this way the dysphagia and the convulsion, hypomyelination means the Schwann cell deficiency, ataxia, and the dysarrhythmia, and the dysphagia and the hypotension also occur in the 1 to 3 years old person, and the gradual more motor cerebral and spinocerebral function loss. So that is the 5 to 9 year old person which that involved this complication. So this is the effect of the hex A, the hexosaminidase A deficiency, which that will lead to Tay Sach's disease. And this Tay Sach disease is basically cause this problem and complication, will lead to cause death also. So the prevention through drug treatment or the treatment combined with the DNA and enzymatic screening program currently in use will lead to eventually control of this disease. So I hope you make sense about the Tay Sachs disease. This was the video about the pathophysiological and drug treatment and the effect of the Tay Sachs disease on the human body. So thanks for watching. If you like this video, please make sure to subscribe, like and share. We will see you with the next video. I hope you make sense. Bye.